welcome to Lab 1. This is our histology lab. So this is our very first lab that we do. This lab, we're going to be looking at tissues. So I'm doing this I have to do this in a slightly different way. I'm doing it in a classroom, so if the audio sounds different, just let me know. If the audio is bad, it will sound a little different. I'm in a classroom, but I wanted to be able to use a classroom that has a touch screen so I can draw on the tissue slides and kind of let you guys know what the things are. So this is the histology section. In lecture, we talk about histology and what it means. So we did talk about that. So as a reminder, if you cannot do great things, do small things in a great way. So as we get started on this lab, these are the things that I'm required to discuss, and I'll be discussing here, and just really getting a little bit of stuff in. So anyway, these are the objectives that we'll talk about. Basically, kind of think of your objectives like a study guide, so to speak. So as we get started, let's get started what a tissue is. What is a tissue? Tissue, all it is, guys, if I take up, so if you remember the hierarchy of life from lecture, that what you have is our subatomic particles come together to make atoms, atoms come together to make molecules, chemicals, chemicals come together to make organelles, organelles come together to make cells, and cells come together to make tissues. So a tissue is a collection of cells, and we can even say cell products, that come together, do a very specific function, and they can create organs. And when they create the organs in the body, they come together to make the organs, like the stomach is made up of multiple types of tissues. And the study of tissues, that term is called histology. Okay, so we can have cells and cell products, like the secretions and then things like that. So cells and their products, they secrete things. So we can say cells and their secretions can come together. The material, the products, the things the cells produce, the proteins that they secrete, the fibers, the fluids, things like that they produce. The cells and the things they produce make the tissues, like cells, something called extracellular matrix, for example, all that comes together and makes tissues. Tissues can combine to form organs, the organs produce the organ systems. Okay, basic hierarchy of life in the tissues. Now, when we're in these tissue section, we are going to be learning some tissues. We'll learn the epithelia, which there are 8 of them that we need to know. There will be 12 connective tissues that we'll learn, 3 muscular tissues, and 1 type of nervous tissue. So there'll be 8, 12, 3, and 1 of these that we're actually responsible for. So there's 4 types: epithelial, connective, muscular, and nervous tissues. And these are the ones we're responsible for. Now, how many we're responsible for, there's the numbers. Now, as we get into epithelia, what we really want to talk about is, if you kind of think about, we said that you can be the cells and the stuff they secrete, okay? So the stuff they produce, their products, things like extracellular matrix, things like that falls in the world of histology. So epithelia, these cells, the epithelial cells, they're layers of cells, okay? The layers of cells that cover the internal or external surfaces. So this covers this inside and out. So this would be your outer surface, like your skin would be epithelia, the lining of your mouth, epithelia, the lining of the anus, epithelium. So epithelia lines us inside and out, covers us an internal and external covering. Now, this also doesn't just include the cells that line us inside and out. It also includes the stuff that produces fluid secretions, and this is glands. Glands are things that produce a fluid secretion, like a hormone or sweat, body oils, that kind of stuff, hormones, etc. Okay? So let's take a look at those. Now, one of the things epithelia does, what do? They have some jobs. Tissues have jobs to do in the body. And because they have jobs to do in the body, they have structure that allows them to do that job. One of the things that they can do is they can provide protection. Now, we're going to see what gives them the ability to do this protection, what gives them that ability. We'll talk about that basically like a thick, warm coat. If it's very thick and layered, then it's going to be really good at providing protection. It can provide protection from abrasion, like we rub up against a brick wall, dehydration, water loss, chemical agents like a chemical that falls on us, an acid or something like that, biological agents, bacteria, viruses, things like that. Okay. Now, they also can control permeability. For example, absorption. They can absorb certain things. So if we look at the lining of the lung, it's really good at absorbing oxygen and things like that. Okay, so it's really good at absorbing. When we look at lining of capillaries, these epithelia are really good at absorbing. So we have that functionality. We can provide sensation. Epithelia have an extensive, or we can say large nerve supply. Sensory nerves are attached to epithelia, so epithelia are really good at that. Most epithelia are really well sensitive, they have sensory abilities. And they also produce secretions like your glands. With the glandular epithelia, they produce specialized secretions like sweat, body oils, hormones, that kind of stuff, okay? Now, to make a tissue, one of the things I want to make a point about these three things is to make a tissue, we got to hold cells together. If we got more than one cell working together, these cells have to come together. They have to join together. They're going to need to stick together, attach to each other. How can they do that? They need cell junctions. Cellular junctions, there are what we call three cellular connections. Cellular junctions, three intracellular connections that are used to hold cells together. Now, at first, all you really have to know is the name and they exist. I will go into some of the details about them just a little bit, more than we need to know. We will talk about these when we get into skin and things like that as well. We'll talk about these especially in skin. Some of these come back at us in A&P2. But I talk about most of these we talk about in skin, or we'll talk about it in the heart. And that's kind of where we'll see these guys again. But we'll start with one called occluding junction. Now, occluding junction is sometimes called a tight junction. Now, when we look at occluding junctions or tight junctions, they are watertight. They prevent leakage of fluid. So they are a watertight barrier. Gap junctions, on the other hand, they are open. They are little tubes that let water and other ions diffuse through them. And then, so they're called gap junctions. Macula adherens, which these others fall into the place of macula adherens, desmosomes, for example. Your desmosomes, they're kind of like spot welts. They use a molecule called a cell adhesion molecule. A cell adhesion molecule holds cells together. This is what allows for multicellularity. If you guys don't know what multicellularity is, that means we're made of more than one cell. That's why we need these. These are for multicellular organisms, need these proteins to hold the cells together so each cell can cooperate. Very important, and that is. So macula adherens or desmosomes. I like to think of these more as spot welds. These are attachment junctions. Occluding junctions, gap junctions, and macula adherens are the three kinds of intracellular connections. Okay, now these intracellular connections, this happens to have a really good image of our basement membrane. Now, in the basement membrane, you can see two of the layers here actually on this. It's the only thing that shows the basement membrane at all. The basement membrane is what is going to be, it's going to kind of, if you've ever seen anybody put down tile flooring, think of the tile, that's going to be the epithelium. Well, the basement membrane is going to be like the backer board that we put down to put the tile on. So this basement membrane is the structure that connects the epithelium to other tissues, kind of like the backer board. Now, this backer board layer, basement membrane, has got a basal lamina and a reticular lamina that makes it up. Now, the basal lamina has got two distinctive sublayers. It's closest to the epithelium. It's actually secreted by epithelium. It has got a clear layer and a dense layer. That helps make part of the basement membrane up. That's not entirely basement membrane. Then there's the reticular lamina, the deepest part of this. And the reticular lamina is secreted by connective tissue. It's the deepest layer. It's called the reticular lamina. So basal lamina is made up of a clear lamina and reticular, clear layer and dense layer, sometimes called a lamina lucida for clear, lucid, and lamina densa, dense layer, okay, densa. Okay, so kind of think of it like the backer board. Now, here we have our basement membrane. Down here is our backer board. Here we have our epithelial cells on it, like our tile. Now, one of the things I like to just talk about very briefly, because you will hear me use this word sometime, is we will see structures on cells. We will talk about the basement membrane, the basal lamina, the basal surface, so to speak, and we'll hear me sometimes talk about the top surface or the apical surface. And I like to give this as just you will hear me talk about it and reference it, the top surface. The top surface has these things on it called cilia or microvilli sometimes that might be there. They're not always there, but they could be there, cilia or microvilli. These are structures that might be up on the top or apical surface. Sometimes cells are long and tall, sometimes they're thin and flat, sometimes they're more boxy. And we're going to see these differences, okay? Now, and that's what we're getting ready to talk about now. Now, because cells can have different shapes, there's ways we can classify them. We can classify them on shape, and we can classify them on how many layers. Now, this is very important as we get started with epithelium. This gets us 6 tissues off the bat. Now, remember, if we go back and we look right here, if we just go back real quick, we have to know 8. 6, 7, 8. That gets most of them already. 6 are going to be gotten by knowing this slide. That gets 6 of the 8. 2 of them will fit this slide, but most of them will fit this slide. Now, we can look at their shape of the cells at the apical surface, or we can look at the number of layers. Now, if we have a cell that is very flattened, an epithelial cell, and this cell is very flat, like look right here. This cell is quite flat. Look up here at the apical surface. These cells are quite flat. Now, though these cells are boxy down here at the bottom, the cells at the very top are very flat. So we have flat cells here at the top here, okay, and flat cell here. That is a word that is called squamous, okay, squamous. So squamous, squash, cells that are flat are called squamous, okay? And if they are squamous, we can see squished. Think squished, squashed, squamous. Cuboid is boxy. Look at how more boxy these are. These are more squared. These cells at the top are more squared. Look how square these are. And look at how square these are. Columnar is long and tall. Columnar is tall. The cells at the top. Now here, if there is, one of the rules is, if you have layers, the bottom cells are boxy, and the top cells take on the shape. The top is flat, the top is boxy, the top is long and tall. Okay? So squamous, cuboidal, columnar. Now that's just by shape. Now let's look at the number of layers. The number of layers that we can have, you can have one layer, like right here, we have one cell thick. Just one cell thick, right here, one cell thick, right here, one cell thick. Okay, one cell thick. Here we have many cells thick, many cells thick, many cells thick. One cell thick is called simple, one cell thick. Okay, stratified is many cells thick. Okay? So you at least have multiple layers of cells here that are in layers called strata. Okay, so simple is a single layer of cells, and stratified is 2 or more. Now, take these together and combine them, and we get 6 new words. We get simple squamous, simple cuboidal, simple columnar. Those are cells with one layer that are either flat, boxy, or tall. Then we can have stratified squamous, stratified cuboidal, and stratified columnar. Those are cells that are 2 or more cells thick, that are flat, boxy, or tall, in order, okay? Now, that's very important that we understand this. Very important that we grab that part, okay? Now, once we got that part down and we understand it, let's move on to the rest of this. Now, one of the big things I always like to talk about is this. That when you have cells like epithelia, they have got to be replenished. Some epithelia, because they cover surfaces, they are lost. They might be around weeks, months or so. It can take like a month for a cell to get from the bottom of the skin to the top of the skin, and then it might be there a couple of weeks, and then it sheds, or a week or so. So cells, they live, they die. Okay, so there's a continued division of stem cells on epithelial cells. These are called basal cells or germative cells. Then these are stem cells, and they're always near the basal lamina. Now that's very important that they're always dividing. This is where we're going to get new cells from. Okay. Now, this is where we're going to constantly get the new ones. This is also why epithelia, because there's continued division of cells, this is why epithelia is always so big for cancers. Okay? So, remember, with the 6, there, the first 6 that we have, they're going to fit our nice little rules that we have. Okay? What we do, we look at first the number of layers, and secondly, the shape of the cells that are on top. Okay? So if it's one layer, then the cells that are on the top is the only cells. Okay? So we can be simple squamous, simple cuboidal, simple columnar. Good. We can be stratified squamous, stratified cuboidal, stratified columnar. Good. We get these 2 tissues that don't match these rules, and that is transitional epithelium and pseudostratified ciliated columnar epithelium. Now, transitions, it's going to have the ability to go from a stretched appearance to a more recoiled appearance. It can transition. Pseudostratified ciliated columnar epithelium. Pseudo means false, stratified. It is falsely stratified. It is not really stratified. Ciliated, it has those cilia we mentioned. Columnar, the cells will look tall and long. Okay? Epithelium, it lines the body surface. Okay? Now, so here's my tips and tricks. These aren't in the lab instruction sheets, but these are my tips and tricks. First thing you want to do, you're going to look for a free surface. So let's jump in the notes, ahead of the notes, and let me show you these application of these tricks on some stuff. We're going to look for a free surface. Only epithelia have free surfaces. Let me show you what I mean by that. Zoom in right here. The free surface will be these surfaces right here, and I'm going to have to erase these that air is touching. Okay? Okay. Now, I'm outlining free surfaces. Like right here's a free surface. Okay? Now, this is a top-down view, so you're not going to see a free surface, but I've outlined the free surfaces. We see that there is a free surface. There are surfaces that are exposed to air. Okay? That is the first thing that tells you this is an epithelium. Okay, so if I see a free surface, we see a free surface where it is exposed to air. Since it's exposed to air, an exposed free surface, that is unique only to epithelium. Next, we're going to see, is there one layer or two or more layers? Then we're going to be able to say it's simple or stratified. Then we're going to look at the free surface and see what the shape of the cells are at the free surface, or the cells at the top, cells near the free surface. What are their shapes? Okay? Now, but I wanted to show you a free surface because students, it's not in the notes anywhere, but this is what I call it, is the free surface. But that is very important if you're going to be able to identify these tissues. The tips and tricks that I give on that is going to be helpful. So our first one is simple squamous epithelium. Now, what are we going to be doing with this is, guys, is for every tissue that you need to learn, there are a few big things you need to be able to do. Now, one of the things is a description is going to help you find it, okay? Now, let's say that a friend of yours was trying to tell you where to find something they left you. They left you some money at your house. And they said, well, I left it here. And they gave you a description of where they left it. That would make it easier for you to find if they gave you a description. Or you're to meet a new person. Maybe you've asked on a blind date. You're going on a blind date. And the person that you're going to meet on a blind date tells you, I'll be wearing a red dress, or I'll be wearing a red sweater, okay? And you want to meet them, and I'll be meeting you here. So they give you a description, what to look for. It's going to help you find. If they gave you a description, this is what I look like, this is what I'll be wearing, you'll be able to find me easier. If I give you descriptions of the epithelium, you'll be able to pick it out easier. That's the goal. So that's what I'm trying to do. Okay. Now, secondly, you need to know where they are in the body. Where are they? Where are they located? And what do they do? What are their jobs? Okay? Now, first things first, let's look at simple squamous epithelium. Now, one of the big things it does, you're going to find it in your lymph vessels, you're going to find it in body cavities. It lines blood and lymph vessels, so it's lining blood and lymph vessels, okay? It lines things, so it might line the stomach, the liver, the gallbladder, and other viscera. And it also is in the kidneys, what we call the nephron loops, okay? Nephron loops in the kidneys, okay? So that's what we're going to find, okay? We're going to find them there, okay? Now, what does it do? It exchanges gases, and it diffuses materials. Now, think of it this way. Simple squamous is the thinnest you're going to get. This is the thinnest, lightest tissue we can have. Think of this as just wearing a single t-shirt. And we walk outside in the cold wearing just a thin t-shirt. And we're going to get very cold very quick because it won't protect us. Okay? Wearing a thin little simple squamous epithelium is going to let stuff pass through very easily, kind of like a thin little t-shirt. Okay? So it's very thin. When we look at it, it is extremely thin. One layer of flattened cells that look like fried eggs. Looks like a fried egg. When you look at it, it has a fried egg. Now this is top down, okay? Now, what I'm going to do with this is, these are textbook images of it. Okay, how it looks like a fried egg. Okay, like a fried egg. Then I put the fried egg on there so we can think fried egg. Simple squamous, when you look down on it, it looks like fried eggs. Now, think about a fried egg. It's very flat. When you look at a fried egg from the side, they're very flat, okay, with a little bump on the top. That's what squamous cells look like. They look like fried eggs. Now, you'll hear the word squamous in cancer, okay? Now, what we're going to do is, I'm pretty sure that this thing usually, so I've outlined the free surfaces, okay? Now, where the free surface is. When we look at these cells, notice they are very flat, okay? I really need to erase these because I was kind of saying here's the free surface. These are thin, flat cells. Now, I've got some pictures that I borrowed from books that show them, like this part right here from the kidney, top right is from the kidney, from a structure called a Bowman's capsule. And I am I'm going to erase all this. I'm going to come back and I'm going to show you now what we're looking for. Okay, there we go. I just had to make sure I remembered. It's been a while since I used these. So, when we look at these cells, they're very thin and flat. Okay? So that fried egg appearance. So I'm outlining the tissue and very thin and flat. Now right here, we can actually see these squamous cells right here. So they're looking very squamous, squished, squashed, squamous, okay? So all this right here, these are all squamous epithelium, okay? So the squish squash squamous, simple squamous, one layer. Now this is one layer here of squamous epithelium right here, top and bottom. These are in the lungs. So right here you can see a free surface, okay? That's air right here. This is the lung pockets, the alveoli of the lungs, okay? Now we're looking top down right here. We can really see the egg shape, the fried egg appearance of these cells, okay? This is a top-down look. Now this is looking at it from the sides right here. These are all looking at it from the side, okay? Laterally, okay? Now, what I want to help you guys, if we look at it right here, you can see these arrows pointing to some squamous cells. This is lining up blood vessels right here. These are blood vessels. These are simple squamous. All this stuff right here, for example, that's muscle tissue. This stuff right here is other connective tissues inside here. But it's this stuff where the black arrows are pointing is pointing to simple squamous in the top left. Top right, we've outlined it. Bottom left, we can really see all that simple squamous. And then this is the simple squamous. The bottom right is just looking down on a simple squamous epithelium. Okay? Now, it will take you a while looking at it before you can start to see what I'm sketching, and then you'll start to see it. Okay? But as you begin to see this, because one of the things I do with my tissue is I do expect my students to be able to look at pictures that aren't in the notes on the task. So the task will have different pictures. I want you to learn characteristics and be able to identify them based on characteristics, not just memorize one pig. I think it really helps that you learn it that way. It makes you stronger. It is harder in some respects at first, but it makes you stronger in the long run to be able to recognize these different kinds of tissues if you're ever going to have to do it again. Okay, so that's how I do mine. Is have you learn characteristics. Okay. Now, so as we look at cuboidal epithelium, simple cuboidal, simple one layer cuboidal boxy shape. So we have one layer of cube-shaped cells. Okay. When you look at it, what you would notice about them is that the nucleus is roughly dead center, where there's about the same amount of space The nucleus is about equal distance, equal height. The nuclei are roughly in the middle there, are roughly equal distance. Okay? Kidneys and glands is where we're going to find this in the body. Okay? And it's going to be functioning in absorption, okay, and secretion in your kidneys and glands. Now, right here we have kidneys. So that is 1 cell thick. One cell thick, okay? And the cells are boxy, okay? 1 cell thick of boxy-shaped cells. All this is connective tissue. Under it. And other tissues under it that we're not going to be worrying about, okay? All right. So let's take a look, and so I think sugar cubes. So here we have them, and what I like to do is kind of describe it like this, guys, okay? So the tissue we're looking for is right here. So now let's imagine you took a toilet paper tube and took it and you looked at the end of it, the open end, okay? A tube, as you look at it from the end, would look like that top left picture, okay? Same thing goes, so we can see that there is 1 layer of cells, okay? And these cells are very boxy. 1 layer of cells and they're boxy, okay? Right here, really good, okay? Now, by the way, free surface right here, okay, that's your free surface. These are your free surfaces, so we know this is an epithelium. Now I look at it and I notice there is 1 layer of boxy-shaped cells, okay? 1 layer of boxy-shaped cells that I see here, okay? Same thing goes here. Free surface. You can see the shape of the cells. Okay? Now, that's looking at the end. Same thing here, okay? Okay. Now, this one, let's say that I took a tube and looked at it from the side, and I cut it so that I can see the edges, the 2 edges. That's what we got here, okay? And here's the free surface. Right there's the free surface. And these are the boxy-shaped cells. And you can see the shapes of the cells there. Okay. Okay. 1 layer of boxy-shaped cells. So these are all simple cumulative. They're all just organized in a different way. Okay. They're forming tubes. So if I had a tube like that, okay, I had a tube that I was looking at like this, a 3-dimensional tube, and I looked at the end of the tube, I would see a circle. But if I took this tube like this and cut it this way, long-wise, I might see something that looks like that. Okay? And this edge right here is what we're seeing right there. Okay? That makes sense. Okay? All right, so I like to tell you what you're seeing. Okay, now, all right, right, simple columnar epithelium. Now, here we're dealing with columns, so I've got like a Greek column right here. These cells, so we have one cell thick, okay? One cell thick, and the cells are long and tall, okay? They're long and tall cells. Long, tall cells, one cell thick, okay? And they're long and tall, okay? So you can see they are one cell thick, and they are long and tall. There might be a cell called a goblet cell found present. Maybe there will be goblet cells. They can be found. We'll see some of the goblet cells. Where you will find this tissue is lining your digestive system, lining your uterus, your stomach, and your intestines. In the uterine tubes even, and in the uterine tubes, there's a very specialized type of this tissue called intermittently ciliated simple columnar epithelium. It is ciliated in an intermittent fashion. There oftentimes will be microvilli present, like right here. These are called microvilli. And what the microvilli do, it's like this. Okay, if I have point A and point B, and I went between, and let's say I had a point A and a point B, and these were the same distance, okay? So I did something a little bit wrong here. Okay, I got point A and point B, and it's the same distance. But instead of doing a straight line, I do this. Clearly, the one on the right covers more surface area. These are microvilli. Okay? Microvilli are there to increase surface area that is available for absorption of things like nutrients. Okay? So, it can secrete some digestive juices. Some of your digestive enzymes are secreted, or it can absorb nutrients. That's what we're going to basically say, secretion, absorption. Secretion of digestive fluids and absorption of nutrients. Okay? Basically, all epithelia, what they do is they protect, they secrete, they absorb. Okay? That's their main jobs. Protect, secrete, and absorb. Okay? Alright. So let's look at these. So some people say they look like these right here kind of look like popsicle sticks. Okay? Little popsicle sticks or little bits of gum. Okay, here's the bottom. Here's the top. Oh, did not mean to switch slides like that. And here's all the cells. Really can see them well. Hopefully now it's starting to come in where you can see it. And you're starting to see these characteristics coming in. Same thing right here. Okay. You're probably starting to see it pretty well, okay? Now, these are all simple columnar epithelium. This is kind of doing more like a zipper right here, okay? But that's still simple columnar epithelium. Right here, we have a goblet cell, okay? We have a goblet cell. That is making mucus. These little white guys right here, these are all goblet cells that we see. Okay, these are goblet cells, these little things right here. Okay, these are all goblet cells that we can see. Okay. Alright, so these, and you can see this on the recording, so it's, that's exactly how, that's why I'm doing this in this room. I have a touch screen that I can draw on and annotate. Alright. So stratified squamous. So we are now out of the simple, we're now in the stratified. And we only have 8 of these, so we got 6 of them. That's pretty good. We got 6 of them done. All right, so let's do stratified squamous. So multiple layers and the cells on the top are flat. Okay? Now look up here. The top cells are very flat, but the bottom cells are boxy. Okay? So we have multiple layers, but the top cells are flattened, and so it's stratified squamous. It looks like a stack of pancakes to me. Okay? Now, the outer layer of skin, your outer skin, lining of your mouth, your throat, the vagina, and part of the anus, the anal canal. Okay? Now, in your skin is keratinized. The other places are moist. Everywhere else, all the other places don't keratinize. Keratinize happens on your skin. That's what happens when you get calluses and corns, is it keratinizes extra. Your skin gets keratinization happens. It keratinizes. We will talk about this in skin lecture, keratinization, okay? So what this does is protection, okay? Think about this. When I poured syrup on this stack of pancakes, the outermost pancake gets all that syrup. The one under it is protected from the syrup, okay? So that those layers, the pancakes on the top have syrup on it. Pancakes at the bottom, they don't have syrup. They're not getting the butter and the syrup on them unless you open it up and pour syrup on each layer. Okay? It's kind of like that top layer, the top pancake gets all the butter and syrup, and the pancakes under it don't get it. Okay? So, layers of flattened cells. Okay? Here is our free surface. Okay? So this is an epithelium. Here we have our, down there, our basement membrane. And look at how the cells at the top look very flat. They're very flat, okay? They're flattening out. And you can see how the cells at the top are starting to flatten out. And then at the bottom, they're very boxy. But at the top, they're very, as it moves up this way, they flatten out, okay? How they're very flattened at the top versus more boxy at the bottom. Okay? Now here, that's a lot of keratin. This is keratinization right now. Okay? Now you can see that these cells, they're basically so dead, they're so thin and flat, you can't really see them. You can start to make them out right here. As they keratinize, this is full of keratin, and this is like my skin right here, okay? This is skin. And that's the only, I wanted to pick that one that was keratinized. I wanted to get one with keratin. Now, usually as we get through this, you start to see these very well, you start to kind of see this pretty well, and it really starts to show out. But you notice that each one of these has a free surface. And only epithelia have free surfaces, okay? Because they line the body inside and out, okay? Now, stratified cuboidal. This is 2 or more layers of cells, okay? 2 to 3 is kind of the way we'll go. And 2 or 3. So, in this case, it's 2, okay? Free surfaces right there, okay? Some people say it looks like stacks of ice cubes. I think it looks like donuts. I think it looks like donuts. I think they look like donuts to me, okay? I think donuts, all right. Now, they are found in sweat glands and mammary glands. The ducts, the sweat glands, and mammary glands, and it's for protection. Okay? So here we have 1, 2, 3 layers. Okay? 2 or 3 layers. Free surface is right there. Free surface. 2 or 3 layers. Free surface right there. 2 or 3 layers. Free surfaces right there. 2 or 3 layers of cells. 2 layers of cells on this one. Okay? Okay? Right, that's what you're looking for. Okay? Now, all this other stuff over here, that's just all connective tissues. Okay? That's just a bunch of connective tissues here that we don't worry about yet. Okay? So, that's just all connective tissues and other kinds of tissues, other tissues that we have in the body. Okay? But that's all connective tissues around it. Okay? So we're not going to worry about those. We're looking for the epithelia. That's where they are. You can see that there's multiple layers of boxy cells. Okay, 2 or 3 layers of boxy cells. Okay, 2 or 3 layers of boxy cells. In some cases, 2 to 3 layers. It can be 2 to 3 layers of boxy cells. Is what you're generally going to see for these tissues. Okay. All right. Now, stratified columnar epithelium, what you're going to see is multiple layers, but here we have our free surface. There's our free surface right there. What you've got to look for is the cells at the free surface take on this shape. The cells beneath it are more boxy. Okay? So we have multiple layers, and the apical layer, the cells near the free surface, are elongated. Okay? But there is multiple layers. Okay? And notice these are elongated, and there's more than one layer. One of my students said this looks like babies in a nursery. They are found in your urethra, the anus, and the pharynx, and it is for protection. Okay? One of my students said this looks like babies in a nursery, the hospital. Right here we have some goblet cells, by the way. But we have more than one cell. Okay? More than one cell thick. And the top is columnar. This one is really good. That's a really good image. This one here is pretty good. Sorry, I've still got this little bit of cough. The recording probably not helping. I got sick at the end of the semester, so I have a cough. The end of the fall. Okay. So. All right. Okay. So what we're going to do is help you see that. Now, transitional. Transitional, so I say Mario Mountains. One of the things I'm also going to do with transitional is think tombstone. We're going to think they look like tombstones, or rest in peace. Okay? They're going to have this tombstone appearance. Okay? This tombstone appearance to them. Okay? Like a tombstone, they rest in peace, transition. They tombstone. Okay? It changes as the bladder empties and fills. For example, when the bladder is empty, it's going to have that curved tombstone appearance. When the bladder is full, it's going to stretch and it's going to look more like a stratified squamous. Okay? And this can shift. So it's found in the bladder and ureters. Okay? The urinary bladder and ureters. What its function is to cycle stretching and recoiling, filling and recoiling. But it has another job. Urine can absorb in it. So I'm giving the tombstone appearance to this as an extra help. It looks like the mountains in Mario's with the curved tombstone appearance to its, so think of a tombstone, okay? So we can see these tombstones. Every one of these, we have multiple layers, okay? And it's got that classic tombstone, okay? These tombstone appearances for transitional, okay? It looks like tombstones. So they tombstones, okay? All right. Very good. Okay, so I hope that helps. Now, my favorite is pseudo-certified ciliate columnar epithelium. Woo, that's a big word. Pseudo, falsely, stratified, falsely stratified. Pseudo-stratified ciliate, it's got cilia. Columnar, long and tall cells, epithelium. Now, not truly stratified, they appear because of very positions of the cells and nuclei, the way they sit. There's cilia, the little hairs that is on it. You will find it in the nasal cavity and trachea. Also found in the ductus deferens, or that is also known as the vas deferens. Vas deferens. Okay. The vas deferens or ductus deferens. Okay, now it is going to protect and secrete because there are mucus secreting cells that may be here. The mucus that it secretes, when stuff falls on it with the mucus, it traps debris, and then that debris gets taken by the cilia, and then it brings it up and we clear it out the throat. It's what we call the mucociliary escalator kind of thing. It's a mucus escalator that brings it up, our mucociliary pathway that brings this stuff up. Very important in cleaning debris out of our airway. Okay, so we can cough up stuff. Okay, now. So as we look at it, we can see that it looks stratified. Here's the basement membrane. It looks stratified, but right here we have cilia. That's the telltale sign. Here we have goblet cell that might be there. Okay. Now, the cells aren't truly stratified. Though they are layered, though they appear to have layers, they're not truly stratified. Because the positions are varied, they are not truly stratified. It is falsely stratified. Okay? So you can see it doesn't fit this columnar. It is pseudostratified. Now the one that has cilia on it, that's pseudostratified. Okay. Sorry, it takes you a few minutes to make that work. Okay. Now that is our last tissue to identify for this part of the lab. Now what we're going to be doing next is to actually look at some glandular stuff, and then once we finish that, we'll stop right here, okay? We'll stop pretty soon. Okay, now, really big things that we're going to talk about. Now that you've got the tissues, one of the things I'm going to tell you that we've seen the tissues, it will take students about 8 times going through these various slides and tissues and getting to look at it before they really start to get it. Like the first time you look at it, it looks like a bunch of blobs. It looks like a bunch of pink and blue blobs that you're like, I don't see any of what my professor says. But it takes a while to start to see it, okay? As you start to see what I'm doing and you start to kind of follow along with me as I draw on it and look at it, it will take various times looking at it before you guys can really see it, okay? And really see what I'm showing you. Okay? All right. Now, so it takes about 8 times. Now, glandular epithelium, they secrete stuff. They release secretions. Now, there are really two kinds of glandular epithelium that are glands. A gland can be exocrine or endocrine. Now, exocrine have ducts. They deliver through ducts. They do deliver products through ducts. Endocrine is ductless. They do not have ducts. They do not deliver products to targets through their duct. They secrete the fluid around the gland, and that diffuses into blood. They make hormones. Endocrine versus exocrine is a big deal. This will be on the exam. There will be a multiple choice question for sure on endocrine versus exocrine. Okay? Okay. Now, how do we secrete? We can classify glands by how they secrete, the mode of secretion. Merocrine, it uses vesicles. Vesicles come out, this is called exocytosis. This is how your sweat glands and salivary glands work. Apocrine, we shed the top. We shed the top. Contents release are shedding the apical portion of the cell and the gland cells, and it secretes it. Holocrine, we kill the gland cell. We kill the gland cell so the whole of the cell dies. Whole of the cell dies. Cells can be replaced because they die. Sebaceous glands, oil glands. Okay? Mammary glands are apocrine. Merocrine glands, they go through exocytosis. Okay? Know these. I will ask these as a multiple choice question. One of these will be a multiple choice question on the test. Okay? Alright, serous glands. Now this has to do with what they secrete. Serous glands make very watery secretions, like things that have enzymes, like saliva. Mucus glands make mucus, and mixed glands make both serous and mucus. Your salivary glands are mixed. They make both serous secretions and mucusy secretions. Okay. And then we can classify them based on how many cells they are made up of. If they are one cell, like a goblet cell, they are unicellular. Goblet cells make mucus. They are unicellular. They're found amongst your interstitial intestines, intestinal lining, and your tracheal lining. Your multicellular glands with more than one cell, they can be simple if they have one duct, and they can be compound if they have multiple branching ducts. So this means they must be exocrine. Okay? So here we have simple glands have one duct, and then compound glands have branching ducts. Okay? All right, this is where we're going to stop. And we'll pick up here in the next one. Thank you, guys, and we'll pick you in the next one.